Hi, my name is Trina and I will be explaining the process of signal transduction through membrane receptors, G proteins, and cyclic AMP. Transduction is the process of taking a signal from outside of the cell and converting it and bringing it inside the cell. The first component in this process is the hormone receptor protein structure. This structure is embedded in the cell membrane and it has 7 transmembrane spanning regions with 3 subunits: the beta subunit, gamma subunit, and alpha subunit. These 3 subunits make up the heterotrimeric G protein. The first step in the transduction process involves a ligand. A ligand is a chemical, for example a hormone or a neurotransmitter, that carries a signal from the outside of a cell to the inside of the cell. However, the ligand cannot pass through the hydrophobic region of the plasma membrane, so it uses the help from the G protein. First, the ligand binds to a metabotropic receptor. After the ligand binds to the receptor, the protein goes through a conformation process and it changes shape, which triggers a mechanical reaction that causes the alpha subunit to replace guanosine diphosphate with guanosine triphosphate. After replacing GDP with GTP, the alpha subunit separates from the G protein and migrates through the cell to another protein called adenylyl cyclase. Before the alpha subunit attaches to the adenylyl cyclase, the protein is inactive. Once the alpha subunit attaches to adenylyl cyclase, the protein can now carry out its function by converting adenosine triphosphate, ATP, to cyclic AMP. This conversion happens because the ATP loses 2 phosphate groups and adds a cyclic portion, creating cyclic AMP. Cyclic AMP is a secondary messenger that spreads throughout the cell and targets protein kinase A. When protein kinase A is targeted, the cyclic AMP binds to the regulatory portions of the kinase, releasing the catalytic portions of the kinase, which then go through the process of phosphorylation. Phosphorylation is the process when the catalytic portions of the kinase gain energy from ATP and now become activated. Once the catalytic portions of the kinase are activated, they can now move throughout the cell and act on other enzymes. To stop the process, phosphodiesterase, which is an enzyme, catalyzes the degradation of cyclic AMP back to AMP, which will later be used at the beginning of the process to form ATP through the process of phosphorylation. After the cyclic AMP turns back into AMP, the alpha subunit then returns back to the G protein and reattaches with the beta and gamma subunits. The G protein and membrane receptor are now ready for the process to start all over again when the receptor receives another signal from a ligand. Through the process of transduction, an outside signal is processed, which triggers a cellular response to convert the signal into a message that will tell the cell what its function is. This completes the process of signal transduction through membrane receptors, G proteins, and cyclic AMP.